Hello, my name is Parvin Singh and welcome to my video entitled as Unconventional T cells and Unexplored Territory. Unconventional T cells are non-MHC restricted T cells and they have been found as a minor subset of the T cell in peripheral blood. They are being associated with the immune-mediated disorders such as autoimmunity, inflammation, infection, allergy, and cancers. Not only their decrease in number, but their phenotypical changes is also being associated with the immune-mediated disorders. In our study, we have identified the invariant natural killer T cells, gamma delta T cells, and mucosa associated invariant T cells on a total of 203 peripheral blood sample of healthy adult Caucasian populations using a multicolor flow cytometric panel. And the total number of the sample have been categorized on the basis of the gender-wise distribution as well as their age-wise distribution. Data as a whole results revealed that INK T cell represents 0.095%, gamma delta T cell represent 2.1%, and mate cell represents 2.9% of the total T cell population. Age-wise distribution results revealed in the figure 2 that INK T cell doesn't shows any statistically significant difference between the age groups. However, the gamma delta T cells and mate cells shows the significant difference within the age groups. In figure 3, the mate cells and the gamma delta T cells shows the negative correlation with the age that shows that they are following immunosenescence in this population. However, in the case of the INK T cells, it doesn't shows any negative correlation with the age group. This is a gender-wise distribution results showing the descriptive statistics of the unconventional T cells, and the data was categorized on the basis of the male and the female. The gender-wise data reveals that INK T cells and the gamma delta T cells shows the significant difference between the gender. However, the mate cells doesn't show any statistically significant difference within the genders. And the similar result we have also obtained in the case of the mate cells and the gamma delta T cells. They are negatively correlating with the age in the case of the male and female both. However, in the case of the INK T cells, they did not show any negative correlation with the age. Uh, Whereas in female group, the INK T cells are showing the negative correlation with the age. In conclusion, the gamma delta T cells and mate cells was higher in younger age population. Unlike the INK T cells, it doesn't show any significant difference within the different age group. And the gamma delta T cells and mate cells supports immunosenescence, while the INK T cell do not. The gender-dependent variation was observed in the case of the INK T cells and the gamma delta T cells, whereas the female shows the higher number of INK T cells than the males, and the gamma delta T cells was higher in the males than the females, and the gender-dependent variation was not observed in the case of the mate cells, and all the unconventional T cells suppose the immunosenescence except for the INK T cells in the male group. The usefulness of this study is to provide a comparative data when the pathological conditions such as cancer and their prognosis and autoimmune disorders can be compared with the healthy population data to see they are increasing or decreasing in these pathological conditions. So once again, thank you so much for attending this.